Good afternoon, before noon, everybody. First, I'd like to express my deep thanks to the organizers, Eduardo, and maybe also to Herbert, who kind of volunteered me to come here to this very nice, beautiful city for a couple of days, which I now learned is almost identical to whole Argentina, if I interpret your slide, Tom. But almost, yeah, that's some of them will. By the way, don't worry, I'm not a soccer fan. All right, so let's jump into it, and I'm very grateful also to Dario to already set the stage for atomistic simulations, which enables me to speed up a little bit in the interest of this session, I guess. And I will just use one protein to introduce you into the method, and then we'll share a couple of stories with you. And the protein I'd like to use to just get you into the method is aquaporin, which is a membrane protein, the only purpose of which is to conduct passively water across membranes, but only water, and at astonishingly high efficiency of about a water molecule per nanosecond, which makes it ideal to study it by molecular dynamic simulations. As you have seen also before, we are limited to short time scale, or to put it a little bit in a more positive way, we have ultra high time resolution, so nanoseconds, microseconds these days. All right, so what you're doing is you set up a simulation system, you put each atom at a position, for example, from X-ray crystallography, into your computer, and then you invoke fundamental physical laws, quantum mechanics, as we have seen before, Newton mechanics, fundamental laws to calculate the forces on each single atom, and under the influence of these forces here, for each atom here symbolized also by kind of interactions and springs, all the atoms, the motion of all the atoms is calculated. So the point I'm really making here is it's all based on fundamental physics law. There is not much way to fiddle around here. You have to obey the physics law. That is something which distinguishes this kind of approach from many other models, which I would call top-down models, typically used in biology. That requires a huge amount of computer power, but once you employ that, and I should say the Max Planck Society generously employed that with quite a bunch of computer power and a bunch of processors, then the raw data which you get looks a bit like that. So what you can see here, the blue is the aquaporin embedded in a membrane, green and yellow, and you can also nicely see the self-diffusion of the water here on ultra high time lapse. About a picosecond is a second here on the movie. You don't see much motion of the water actually here, so the water molecules seem quite immobile, simply because the time is too short. If we give a little longer time and wait for a month for the simulations, then you can see nicely here this yellow water molecule traversing the channel and ending up on the other side. And it was already mentioned by Dario that, by the way, I couldn't underscore your key points at the last slide more, very important, so one has to be really careful interpreting things. So here you can simply count the number of water molecules which you see past the molecule, the protein, during your simulation, and relate that directly to a measured permeation rate, and it turns out that it agrees to about 20%, a factor of 1.2, which is really extraordinarily accurate. And we could also went one step further and predict a couple of mutations on the basis of an analysis of the energetics, and I don't have time to go in there, just suffices it here to say, on the basis on seeing what interacts how strongly with what during water passage across the channel, we now understand very well how this channel can be so efficient and how it can be so selective. The selectivity, for example, comes about by the protein generating a high barrier, electrostatic barrier, against ions, which lets dipoles like water molecules pass, but not ions like H3O plus or calcium ions or others. So this we understand nicely on the basis of equilibrium simulations. And the topic I would like to share with you today is, what do you do if the process under investigation is much slower than the, say, several hundred nanoseconds, which you can directly access by equilibrium simulations? And the examples I would like to discuss with you are, depending on time and depending on the mood of the chairman, 2 or 3 systems. The first being Kinase, which is a putative and now established force sensor in the muscle cells. Then we'll turn our attention to ribosomes, which I guess you all know much better than me probably. And finally, if time permits, we look at whole viruses. So what brought us into that field is the wish to mimic, to simulate atomic force microscopy experiments. They were able, during the mid-90s of the last century, to separate by force, by pulling at it, for example, like shown here, a ligand-receptor complex. However, despite the name microscopy, they weren't able to see anything, just to measure the force acting by the atomic force microscope, the cantilever, onto this yellow here, on the yellow molecule, and to measure the force. Now, this measuring force we can also do. You see this here in the curve in the simulation. So at each instant of time, we know how strongly we are pulling here with the symbolized spring on the right side. And on top of that, we can see what actually happens. Turns out that this unbinding process, which you can see here, is a quite complex sequence of intermediates, for example, in transient interaction with this loop, etc., etc. So that we can see on top of calculating the force. And again, the same story. First thing is to compare with experiment. In this case, very favorably. The force we calculated is actually the same that was measured in the experiment. And then we can go one step further and look at things which are not seen in the experiments. And that process here of pulling at things, like a child pulling at a gadget when it wants to learn about how this thing works, proved very, very successful in the past 15 years and is now known as force probe simulations, or also steered molecular dynamics, as we have seen also in the previous talk. Nicely applied. So more recently we applied that, as I said, to a muscle protein. In fact, titin, which is this green kind of worm here in the whole muscle machinery, which you know all very well, actin, myosin, crawling across each other. And this green titin here keeps the whole thing elastically together, prohibits it from falling apart. And in order to do that, it has to be elastic, and it also has to absorb two large forces, so it acts as a shock absorber, which was nicely shown in AFM, atomic force microscopy experiments by Hermann Gaub and Julio Fernandez and also others. You can see the basic principle here. So that would be a construct consisting of 8 Ig domains. The whole thing, I should say, is built up by a whole lot of these Ig domains, a huge protein symbolized here by these beads. And when you pull with the cantilever here, then you can see one after the other of these Ig domains being unfolded. And this unfolding leaves a fingerprint in the force. The force drops dramatically, and then the next is being stretched up to a point where it unfolds, etc., etc. So that gives this nice sawtooth pattern. 2 minutes late. So from this sawtooth pattern and from fitting what is called a worm-like chain model, are these curves here, you can extract the length of the part which is being unfolded, the length of the unstructured part, like this one here. I'll need that later on, so please keep that in mind. Now, in all these chemically completely inert and mechanically active Ig domains here, Matthias Gautel discovered one single one, the red one here, which is a kinase, so which shows enzymatic activity. And he solved the structure, and it turns out it's a typical kinase, the task of which is to phosphorylate a substrate, which is just typically the start of a signal cascade. And this, except for this inhibitory alpha helix, which is right blocking the active site here. And the idea quickly came out, well, if we pull at both sides, as it happens in the muscle, then maybe this beta sheet here gets unfolded, destroyed, and then maybe this alpha helix here gets retracted removed out of the binding site, which would in this case activate the enzyme, which would say, well, that is a force sensor. Now, is this all true? And there were raised quite a bit of doubts in the community, particularly because the whole thing couldn't be expressed as a truncated version without the alpha helix. So people thought maybe it falls apart immediately after removal, in which case it couldn't act as an enzyme. So we started out to pull at things in the simulation, like shown here on both sides here. And as you can see, the red helix here in the surface plot is very gently removed out of the blue active site, the dark blue active site. And a closer analysis reveals that the site is really virtually unperturbed by this quite severe action here. And in fact, we were able to calculate the unfolding force, and again, it agreed nicely with the experiment. And there's one nice side story to it. There are two beta sheets here. The one which needs to be destructed very early in order to be able to activate the enzyme. And the second one here on this side, on the N-terminal side, which needs to be intact to preserve and protect the active site. Now these two are very similar and have the same unfolding free energy, and yet, as it turns out from the simulation, this one unfolds very early and this one unfolds very late. How can that be? And that's a nice story about energy and force and the difference between the two. Physicists know that very well. You can have high force, low energy. You can have low force, high energy. They are not necessarily correlated, and nature exploits that here very nicely to make this molecule function. Now, is that again all true? That's all very nice. But the next thing which these simulations suggested were a nice experiment. Now, if we pull in the experiment, and this was a collaboration with Helmut Grubmueller in this case, and Matthias Gautel, if we pull here, the protein unfolds stepwise, and again we have this sawtooth pattern in the AFM. Now the idea is, if we put ATP at an intermediate stage, we pull a little bit, we open the active side out, and then we put a little bit of ATP into it. If it binds, that creates additional interactions, and that should modify this fingerprint here, if it binds. So that would be a proof that actually the binding site gets opened up transiently, as we have seen in the simulation. And indeed, you can see an additional peak here coming up, which shows indeed ATP does bind. So that's a very nice prediction which we made and which was confirmed here later by AFM. The question, of course, came up, and that brought the ball back into our lab. What are these interactions? What happens when ATP binds, and how is the unfolding affected? So we started to buy a couple of new computers and do the whole unfolding pathway, and I don't have the time to go into detail here, but you can see the unfolding of the protein in the presence of ATP, and we compared that to the unfolding in the absence of ATP. And the bottom line is, besides that we now have a very detailed picture of what happens during unfolding here on the left side, if you look at the forces here with ATP and without ATP, they are very similar, except in this region where we have a significant difference, which in fact is the one which gave rise to these force peak here on that part here. And a quantification, a quantitative analysis here on the lower side in the experiment, black without ATP, red with ATP, and here from our simulation where we had studied the unfolding, shows a one-to-one agreement which establishes, number one, that our simulations are quite close to reality when it comes to unfolding, and number two, that indeed, yes, we do understand now very well that titin kinase can act in the muscle and does act in the muscle as a force sensor. How much time do I have? 5 minutes for the ribosome. Actually, I thought that would be my main part here. That is a close collaboration by Holger Stark, who was already mentioned, by Rainer Luhrmann and Marina Rodnina. And to briefly get you into the ribosome, for those probably physicists who don't know what that is, we have two parts here, large and a small subunit, which associate on the messenger RNA, and shown here as a red line. And the tRNAs here, two of them are shown here, read off by specific binding of the codons, the amino acid information from the mRNA, and on the other side they carry the right amino acid fit into this codon, corresponding to the codon on this side, and by moving through the ribosome, prolong step by step the nascent protein according to the genetic code. Now there are in fact 3 binding sites here, A, P, and E. 2 of them are occupied here. And the question was, how do these tRNAs here travel from the right side and leave on the left side? How do they travel through the ribosome? And subsequently I will denote this in this symbolized way, the 2 tRNAs typically here traveling through the binding sites. Now this groundbreaking work of Holger Stark here enabled us now for the first time to see kind of a movie, how this traveling occurs, how the motion or the translocation of the tRNA occurs. He was able to sort several million single cryo-electron microscopy particle images into like 50 classes, group them together, average them together, and come up with resolution, quite remarkable resolution structures for each of these states here, which are shown here. Now, this is not yet atomistic resolution, but what we did now in collaboration with Gunnar Schröder is to take an X-ray structure, which is only present for this very one stage here, and fit it into the electron density of each of these, in this case, 20 selected structures Holger provided. That gives you an impression how that works. Now, this is the X-ray structure, and you see that it fits nicely into the much lower resolution electron density. So at the end of the day, we have like 20 near atomistic resolution structures for 20 subsequent steps during translocation. And this is what we get out from that. So you can nicely see here how playing back and forth, how the tRNAs go through the ribosome, and you can also see the huge motions, for example, the head and the 30S and 50S, how they swivel against each other. And I would just put your attention also to this part here, which is called the L1 stalk, which you see nicely co-moving with the tRNA. And here immediately the question comes out, what pushes what? What's the driving agent? Does, like it appears here, the L1 grab this orange tRNA and pull it out of the binding pocket, or is it the other way around? Does the tRNA being driven by something else and pushing away the L1 stalk? This you cannot see from this sequence of snapshots, and you have to go into simulation and into forces to study these. All right, first question, is that all true? Are the structures which we get accurate enough? And we were quite lucky because quite recently, after we have done all this fitted, invert, and submitted, there was a new structure now in this what is called the pre-force position here, which we haven't used for our fitting work. We have only used that. And as you can see by comparing the structures, that one shows indeed the lowest difference here to all of our models which we came up with. That's the red line in contrast to the other structures here. So that establishes we were able to predict from the combination of X-ray and cryo-lab data very nicely this structure to an RMSD of 5 angstrom for the experts. That's for such a large system a remarkably low RMSD, and it's at the same order of magnitude as with the initial start, which we have used initially for our fitting. Now, from all that, we were able to carry out simulations for the experts, all fully solvated with all the ions, etc. A huge system of about 2.2 million particles, and from each of these states which I've shown you, we spawned 100 nanosecond simulations. So we have now 2 time scales. We have the very slow time scale spanned by the EM data, corresponding to the turnover rate, several per second, and we have the fast time scales from each of these, and we can learn something by combining those. The first thing is we can now have a close look at the motions. And since time is advanced, I just want to put you again to this part here where we see the L1 stalk moving towards the tRNA. Here it's shown again here schematically, so this moves backwards here, that's this part here, and then co-moving here together with the motion of the tRNA. That has been, of course, already known from the EM, but what the molecular dynamic simulations now add is the fluctuation of the high timescale motions, and they are indicated by the bars. And the important message, in particular, if you look here at the large collective motions between the head and the body of the ribosome, these motions are unexpectedly fast. So already on nanoseconds, we have covered the full amplitude of these motions, and we have a big overlap between adjacent states of these motions, which means that the system can travel between those steps where we have overlap already within 100 nanoseconds. And you can go further, and you can enhance statistical mechanics to estimate the rates which correspond from the simulation, which correspond to these jumps between the different states, and we have done that. And that is summarized here for the 5 motions which I have shown so far. 3 of them, the motion of the tRNA and the L1 here, shown here. 2 of them, the large-scale motion here. There's a lot to be learned from that, but the one which really excited us was that there's a continuous line here for those motions, meaning fast transition. Thick lines, fast transition, thin line, slower transitions, no line, very slow transitions. All of them are very fast, quite in contrast to the tRNA. There we see no lines here and no lines here, which means that of all these mechanical motions which have to be going on during translation, was there a stop thing? Okay. The time limiting step actually is the tRNA. And the question is, how does the tRNA do that? And I'll just show that we now can analyze that in quite detail by analyzing all the interactions between the different involved parts when the tRNA move from here across this chiasm here to the other side of the L1. And in each case, we can now quantify the interaction enthalpy between these states. And one thing which comes up is that the ribosome has invented at least 3 tricks to overcome the barriers between this